The story about my work begins with the influence of a postdoc. When I was a student, there was a postdoc at the Weizmann Institute who introduced me to the idea of antibodies and cloning the cells that make the antibodies. Norman Kleinman. I remember asking him at the time, who cares about this? Why do we care about monoclonal antibodies? He gave me all the good reasons why he cared about it. He wanted to figure out how the immune system works. I was interested in analyzing cancer and realized that these monoclonal antibodies might be a good way of figuring out something about the human cancer cells. So that was 1967, actually. Then in 1973, when I went back to be a postdoc at the Weizmann Institute, I started working on a target that I thought would be a good target, which every clone of human lymphomas or leukemias makes that's unique to that clone, that being the antibody that it knows how to make. So I thought the antibody product of each clone would be a good target for the monoclonal antibodies. And immediately when I started at Stanford, we got cells from our patients from the clinic here, and we immunized mice with the leukemia cells. And then we did Norman Kleinman's spleen fragment culture system, and we got some really good antibodies that would tell the difference between the cancer cells and the normal cells from the same person. And that was great. We could analyze targets with these antibodies. The problem with this technology is that those spleen fragment culture systems don't last forever. They produce small amounts of very high quality antibodies, monoclonal antibodies, but they die. They're not immortal. And that very same year, two scientists in England, Kohler and Milstein, invented a technology called hybridomas. Hybridomas are ways of immortalizing the antibody-producing cells, making them live forever, capturing them by fusing them together with a cell that lives forever, and then cloning the fused cells one by one and expanding, and it produces the antibody that the donor cell knows how to make. And it has the property of the fused cell and lives forever. So I immediately set up the hybridoma technology in our lab here at Stanford, and we made the first monoclonal antibodies against human cancer.
I wonder if you might tell me about when you sort of first realized that this was going to work. I think it was in 1976.
I talked about the target that we wanted to go after, and I knew it would be unique to every lymphoma cell. And originally, in order to immunize the mouse, we had to have that target. We had to have it in quantities, pure. And what I realized is that we can make a hybridoma between the tumor cell of the patient and the Kohler and Milstein fusion partner itself. And that hybridoma would secrete the antibody that the tumor cell knew how to make. That would give me a way to get it pure and get it available so I could immunize mice and make monoclonals against it. And the first time I did that experiment, it worked. It was Thanksgiving Day when the gel came out of the X-ray film developer. The bands on the gels defined the secreted products of the hybridoma that I had made. They were radioactive and they were separated on this gel and then exposed to the X-ray film. And I could see the bands on the gel. And indeed, the hybridoma I had made actually secreted the proteins that I wanted to capture. My whole life for the next 10 years, this was in front of me because I saw the result. The problem for me was that it was Thanksgiving Day and there was nobody around. So I went running up and down the halls. I wanted to show this to somebody. I couldn't find anyone to show it to. I had to wait a day or two before I could show it to somebody. We made monoclonal antibodies using hybridoma technology against the target that was unique to every patient's tumor. And we actually made against one of my patients, whose name was Philip Carr, and we studied it in the lab against his cancer cells. We had done this in mice also, against mouse cancer cells, mouse lymphoma cells, and shown how it could be used to treat the lymphoma in the mice. We made these antibodies, we grew the hybridomas, we purified the antibodies, we put them in the freezer, and we waited. By the time he started not responding anymore to the usual treatments, we actually treated him with the antibody we had made for him. This was a mouse antibody. It was against the human lymphoma target. And we actually gave him this mouse antibody. Mr. Carr had a miraculous remission of his tumor. We published it in the New England Journal of Medicine, and that opened the field of monoclonal antibody therapy for cancer.